Good afternoon, everyone. Welcome to today's class. I hope I'm audible and visible. So, yeah, thank you, Ryan. So, in the last class, we had moved on to a new module dealing with leaf scale water relationship, and we learned about some initial concepts required to get a more understanding of the leaf scale water relationships. So, one of it is what is the physiological process called transpiration. And associated with transpiration, we talked about relative humidity and reading a psychrometric chart that helps us in obtaining all the required values associated with humidity and temperature. And we also began with what is called the stomatal conductance. So we see that stomatal conductance is a process that is going to influence both the photosynthesis as well as transpiration rate. It is the opening and closing of the stomata that is resulting in some amount of carbon dioxide being taken from the atmosphere and some amount of water being lost back into the atmosphere from inside the leaf. And so we can define stomatal conductance as the rate of either carbon dioxide entering or water that is exiting the system. And this is all not only a function, it is a function that is based on how largely the stomata is open or closed, and also based on the diffusion of gases between the atmosphere and the inside of the leaf. So we saw this diagram where there's a schematic which is talking about the carbon dioxide that is entering from the atmosphere into the leaf and through the stomata, and water exiting the leaf again through the stomata. So there are various factors that are controlling or influencing stomatal conductance. One is the outside conditions. What is the concentration of carbon dioxide outside the leaf, on top of the leaf? What is the humidity outside the leaf? Temperature, light, all of these factors are impacting stomatal conductance. Then again, within the plant, there are certain hormonal signals that are triggered as a result of stress conditions that are experienced by the plant, and this also sends signals to the stomata to open and close based on what kind of stress it is experiencing. And then again, as we go from the atmosphere to the plant, to the bottom of the plant, which is bottom part where the plants are firmly seated, and the soil, the plant is obtaining its water from the soil. The roots are taking in water, so if there is no water in the soil, it is also going to induce some kind of stress to the plant, in response to which the stomata is going to open and close. We also talked about the leaf water potential, which is basically the potential of water in the leaf that is resulting in the entry of water inside the guard cells of the stomata, that is going to again govern its opening and closing. And then we looked at these two equations, very simple equations that represent A, that is assimilation rate or photosynthetic rate, and T, which is transpiration rate. So A is proportional to the stomatal conductance as well as the concentration gradient of carbon dioxide within the leaf and outside the leaf. In the same way, transpiration is also dependent on the stomatal conductance and the vapor pressure deficit. Vapor pressure deficit is nothing but the difference in the vapor pressure outside the leaf and within the leaf. And whenever we do estimations of vapor pressure deficit, the vapor pressure inside the leaf is always considered as saturated. So we assume that the water condition inside the leaf is the water molecules are saturating the inside of the leaf, as a result of which the water tends to move out into the atmosphere where the vapor pressure will be less. And we looked at what is relative humidity and what is absolute humidity. So when we talk about relative humidity, yeah. So absolute humidity is the actual condition of the water present in the air. So it can be again, humidity is the accounting of the water vapor. So what is the actual uh, what is it, moles of water vapor in the air, or what is the pressure of water vapor in the air, is what is given by absolute humidity. Now, when absolute humidity um, can be described in units of gram per volume or gram per kilogram or mole fraction and or units of pressure. So in our case, we are going to be dealing with mole fractions because we want to know what are the number of particles that are entering the leaf, the moles of particles that are entering the leaf, because we are dealing with it at a micrometer scale of the stomata. The stomata are very small, tiny, around my very 10 micrometer, in the range of some micrometers. So we cannot actually look at a huge mass or a huge volume of vapor that is entering. If the system is very large, like if it is a tank, we can estimate uh, the water vapor in terms of its pressure or in terms of its mass. But since our a scale of measurement is very molecular level at the scale of the stomata, which is at at the sizes of some micrometers. We have to deal with how many moles of water or how many moles of carbon dioxide is going inside and coming outside and all. So that is why when we consider the unit of humidity, absolute humidity, we consider it in units of mole fraction. Now, even though we have to consider it in that, the actual data will be available to us only in terms of this gram per kilogram or gram per volume. So we will look into how we can convert it into the required units that we need. So absolute humidity, like I earlier said, is the actual amount of water vapor that is present in the atmosphere. Now, when it is expressed in terms of grams per kilogram of air, it is also called specific humidity. So if you see the term specific humidity, and if no unit is given, it means that it is grams of water vapor present in some kilogram of dry air. So that is what specific humidity means. In general, it is absolute humidity, but absolute humidity can be expressed in a variety of units, like I earlier mentioned. But if it is expressed in terms of grams per kilogram of dry air, it is called specific humidity. Now, the percentage of this absolute humidity to the saturated content of water vapor in the same air is called, expressed in terms of percentage, is called the relative humidity. And a relative humidity is estimated as a fraction of the actual vapor pressure or absolute humidity or specific humidity divided by the saturated condition at that particular temperature. Relative humidity is different at different temperature. So hence the capacity of air to get saturated with water vapor varies based on the temperature as well as the pressure at that particular point. So all these values are variable with respect to temperature and pressure. So in order to obtain the values of these at varying temperature, we have available some tools, one of which is called the psychrometric chart. The psychrometric chart is helping us estimate the actual vapor pressure, the saturated vapor pressure or absolute humidity, and saturated levels of humidity, the relative humidity at different temperatures, and using the formula RH is equal to actual by saturated into 100, we can get the values of the actual or saturated or relative humidity for a particular temperature. So we also did some problems uh, that would have hopefully helped you in working, I mean, practice, I mean, how to read this chart, like the horizontal lines are corresponding to the absolute humidity. The curved lines are corresponding to the relative humidity percentage. The bottom axis, horizontal axis, is the temperature. And this line, this curved line you see here is corresponding to 100% relative humidity. And you can also see some values along this curved line, which is the wet bulb temperature that should be read diagonally. So all these diagonal values are the wet bulb temperature. The wet bulb temperature at relative humidity 100% is called the saturation temperature. These horizontal lines are giving us values of the humidity in grams per kg, or whatever is the x-axis is unit. So here it is grams per kg. So this is specific humidity. And then when we correspond, when we project these lines to a particular humidity value, we get what is the humidity, actual humidity at that particular temperature. So we also did some problems that would have helped you to read this chart. And we went through the dry bulb temperature is nothing but the actual temperature, normal air temperature. Wet bulb temperature is the temperature to which it is lowered as a result of evaporative effect. Then there's a saturation temperature, which is the wet bulb temperature at 100% relative humidity. Dew point temperature is the temperature at which condensation will start taking place, and we know about relative humidity. So we looked at the relation. We understand that this term is now actual actual humidity or absolute humidity, and this is the saturated level of the humidity. So the difference between the saturated vapor pressure and the actual vapor pressure is what is called the vapor pressure deficit. So we looked at how the variation now for transpiration to occur. I said the VPI is always going to be high in the saturated level. So if this portion inside the leaf is saturated with water, then the only influencing factor over here is the vapor pressure in the atmosphere or outside the leaf. So when this value VPA reduces, what happens is this VPD will increase. So high VPD means the actual vapor pressure is low, and this is corresponding to a low RH, because we know the relation between uh, we know the relation between uh, we know the relation between RH and actual vapor pressure. So it's VPA by VPI. So if VP reduces, RH is also going to reduce. So low RH will result from a high VPD. Now, if this value is very high, what will happen is more water will keep going outside, because this is at a higher value and this is at a lower value. So water will keep going there in excess. So this is going to as a consequence of high transpiration rate, the stomata is going to reduce, because it will understand that there's a lot of water loss. So stomatal conductance, as a response to this high VPD, will uh, start closing up, and then it will try to reduce the amount of water that is going. The same case when VPD is low, the actual vapor pressure is not, it is closer to the saturated condition, or it is lesser, but it is closer. So the conditions of high relative humidity is existing in the environment, and under such conditions the transpiration rate will be low. So because of this, the stomata is not going to be very responsive to it, because whatever is the transpiration rate is optimal. So at this level, photosynthesis will also occur efficiently. Transpiration is not resulting in a heavy loss of water. So as a result of which, it will be in an optimal condition. Now, transpiration, although we talk about it in terms of losing water, is a process that helps in maintaining the temperature levels of the leaf. So, transpiration is not a bad process, but again, the plant has to do some optimization to control the amount of water that is exiting the plant, as well as to keep the photosynthesis process from going on, because otherwise, because again, that is what is resulting in the manufacture of food for the plant. But excess of water cannot be lost. transpiration should happen, but it should not allow it to an extent where the plant is going to lose water immensely, and it is also having to balance the carbon dioxide concentration uptake. Now we talked about stomatal conductance. So one doubt that that might keep coming into our heads is regarding the actual definition of stomatal conductance. So stomatal conductance is a rate, just like how transpiration and photosynthesis is, it is a rate. But we also say that it is the opening and closing of stomata. So how can we represent stomatal conductance with respect to photosynthesis and with respect to transpiration? So stomatal conductance, like I had earlier defined over here, is a rate of carbon dioxide entering or water exiting. So which out of it is stomatal conductance? So stomatal conductance is a process that can be represented in terms of carbon dioxide entering as well as water exiting through the stomata. So when transpiration is occurring, we know that transpiration is a product of the stomatal conductance and the vapor pressure deficit. Now here you can see that I have named this stomatal conductance as GSW. That means that stomatal conductance to the movement of water. Now we are segregating the two processes. So one is stomatal conductance to the water molecules. Here A is the assimilation rate or photosynthetic rate, which is a product of the stomatal conductance to carbon dioxide and the concentration gradient of the carbon dioxide between atmosphere and what is inside the leaf. Now we know that um, carbon dioxide molecules are bigger than water molecules. It has a molecular weight of 44 grams per mole, a molar mass of 44 grams per mole, and water molecule has only 18 grams per mole. So the rate with which the carbon dioxide will be entering will be different than the rate with which water will be exiting the system. Now there is only one stomata. The stomata is open, and through that stomata, water also has to go out, carbon dioxide also has to come in. So in order to take care of this process, we have to somehow standardize this stomatal conductance. We have something called the diffusivity coefficient. So diffusivity is nothing but it is describing how quickly the molecules will move from a region of high concentration to a region of low concentration. So in the case of carbon dioxide movement, the region of high concentration is outside and the region of low concentration is inside. So the carbon dioxide is going to go inside. In the case of water vapor, the concentration of water vapor is higher inside the leaf and lesser outside the leaf. So it is going to go out. But again, like I said, water is smaller than carbon dioxide. So the rate with which, or the rate of diffusion with which, the speed with which this water molecule will go out and carbon dioxide will enter are different because of the size difference. So for this, the diffusivity of water vapor is found to be nearly 2.5 into 10 to the power 5 meter square per second at 25 degrees Celsius and 1 atmosphere at 1 atmospheric pressure, and a diffusivity of carbon dioxide is found to be 1.4 into 10 to the power minus 5. Again, because this one has got the rate with which it is going to be lesser in case of carbon dioxide and more in case of water vapor. Now, we say that carbon dioxide through the same stomata, say suppose stomata is just one opening, and through that opening, carbon dioxide will diffuse into the leaf 1.6 times slower than water exiting the system. So this can be found out by this law of diffusivity by Graham's law, which connects the diffusivity with the molar mass as both of them being inversely proportional to the root of the molar mass. So diffusivity again is a concept that everyone, most of them must have learned regarding the diffusion of gases. So here in our case, we are concerned about the diffusion of carbon dioxide and H2O. And here by Graham's law, we get to know that diffusion or diffusivity coefficient is inversely proportional to the molar mass of the gas. So in that case, when we solve for carbon dioxide and H2O, we get that the diffusion coefficient of H2O to the diffusion coefficient of CO2 will be equal to the molar mass of carbon dioxide divided by molar mass of H2O, the whole root, from this Graham's law. And we know the molar mass of carbon dioxide is 44, molar mass of H2O is 18. And when you substitute these values over here and solve this, you will get something around 1.56. So it is nearly 1.6. So that is why we say that carbon dioxide is diffusing inside the leaf 1.6 times slower than water because of the change in the molar mass of these two particles. So this again can be translated to the stomatal conductance. So stomatal conductance is basically how easily the gases can move through the stomata. Since water has got a higher diffusivity than carbon dioxide, we know that water is a smaller molecule, carbon dioxide is a larger molecule. So water has got higher diffusivity. We say that the stomatal conductance towards water is 1.6 times the stomatal conductance to carbon dioxide. This means that for every, say if you're taking 10 units of carbon dioxide entering, 16 units of water is exiting. So for a particular value of photosynthesis, if we are going to lose 1.6 times the same concentration of water vapor with that rate of stomatal conductance. So if we can so if the stomata is say open like this and it is 5 micrometers, we can say that if the stomatal conductance is say GSC is equal to 10 for carbon dioxide, then GSW is equal to 16 for to water. So from the same stomata, when 10 moles of carbon dioxide enters, 16 moles of carbon dioxide will exit the system. So based on this relation between the diffusion of carbon dioxide and diffusion of H2O. This one value is very important. The diffusivity coefficient of H2O to CO2 is always considered as 1.6, and this relationship is used in multiple regions when it comes to estimating the total conductance, the boundary layer conductance, and so many types of conductance that is resulting in these gases to be entering. So before even we look into the gases at the interface of the leaf as the interface, the gases has to pass through some boundaries before it reaches the leaf. So during all of that, this diffusivity coefficient will be considered in terms of ratio of carbon dioxide water to carbon dioxide. It is equal to 1.6. So So we found out that the ratio of the diffusivity ratio of diffusivity of H2O to CO2 is 1.6. Now in this equation, the vapor pressure deficit, like I said earlier, will be expressed in terms of millimoles of water per moles of air. Because again, we are concerned about the moles of water and carbon dioxide entering. So this vapor pressure deficit will be expressed in terms of millimoles of water per moles of air. And the carbon dioxide gradient will be expressed in terms of micromoles of carbon dioxide per moles of air. Now, when transpiration, as a result of this, when we get the units of transpiration, it will be in millimoles per meter square per second, and assimilation rate will be in micromoles per meter square per second. So, whenever we represent the rate of transpiration to some value in the x-axis, the unit of transpiration is always going to be millimoles of water per meter square per second, and unit of assimilation or photosynthesis is going to be in the units of micromoles per meter square per second. So will be the vapor pressure deficit, which will be the vapor pressure will be expressed in terms of millimoles of water per moles of air, and carbon dioxide concentration will be expressed in terms of micromoles of carbon dioxide per moles of air. So if we solve this equation with the units alone, we see that yeah, we see that if transpiration is in millimoles per meter square per second, then and vapor pressure deficit is in millimoles per mole, this millimole and millimole will get cancelled, and then GSW will be obtained in the unit of moles per meter square per second. So GSW is nothing but the stomatal conductance towards water. So from this calculation, here I have converted millimoles into moles, that is by multiplying multiplying it with 10 to the power minus 3. So 10 to the power minus 3 moles per meter square per second is equal to the stomatal conductance into vapor pressure deficit, which is in again moles per uh, moles per mole. So this moles per moles will get cancelled. 10 per 3 and 10 per 3 will get cancelled. And finally, we will get the unit of stomatal conductance to water in terms of mole per meter square per second. So from here, we are understanding that the unit of stomatal conductance to water will be moles per meter square per second. Now, in the same way, if you calculated for assimilation also, the assimilation is expressed in terms of micromoles per meter square per second, and the concentration gradient is expressed in terms of micromoles per mole. So here also we will get moles and the micromole and the mole getting cancelled, and you will get the stomatal conductance to carbon dioxide is also in terms of moles per meter square per second. So we understand from here that the units of transpiration will be in millimoles of water per meter square per second. Unit of assimilation will be in micromoles per meter square per second, and stomatal conductance, irrespective of whether it is towards water or whether it is towards carbon dioxide, will be in units of moles per meter square per second. So knowing the unit is necessary because again in modeling there will be a lot of estimations. So you have to substitute the quantities provided to you in the using the right units to obtain. We ultimately when you do the calculation you'll get some numbers, but what is going to be the unit depends upon understanding what each quantity is going to be, what unit each quantity is going to be represented in. So transpiration in millimoles, uh, assimilation in micromoles, and stomatal conductance will be in moles per meter square per second. Another unit conversion that we need to look into is the units of carbon dioxide. So carbon dioxide concentration is generally represented in terms of ppm, that is parts per million. It is nothing but parts per million means it's 500 parts of carbon dioxide per million parts of air. So 500 parts of moles of carbon dioxide per million moles of air means 10 to the power 6 moles of air. So that will be ultimately 500 micromoles per mole. So if you get a quantity, say we represent the concentration of atmosphere. So as of now, the carbon dioxide concentration in atmosphere is around 420 ppm. So when we say 420 ppm, it means that it is equal to 500 micromoles per mole. Now, we had looked at the psychrometric chart, and I did say that the y-axis is going to be in terms of grams of water per kilogram of dry air or grams per volume. So under such conditions, how do we convert it into mole fraction is what we look into now. So based on this, I'll be giving a problem. So maybe it would be helpful for you to uh, listen to it carefully. So we know the molar mass of water is equal to 18 grams per mole. Molar mass of dry air is equal to 28.96 grams per mole. Approximately, this is a value that is usually considered. there might be small variations in this value, but again, 28.96 gram per mole will be the molar mass of dry air. Now, suppose we are getting a value of say 10, say 10, 10. This is the value that we are getting, grams of, for actual vapor pressure. So, in order to convert that value into mole fraction, what we have to do is for one mole, if 1 mole of water consists of 18 grams of water, then 10 grams, 10, yeah, the number of moles in 10 grams of water is going to be 10 by 18 moles. That is around this much value. This will be the value for 10 grams. In the same way, we will also find out the number of moles in dry air. So if 1 mole is going to have 28.96 grams of dry air, then 1000 mole, 1 kg, that is 1000 grams of dry air is going to again have 1000 by 28.96, around 34.53 moles of dry air in 1000 grams. Since it is per kilogram of dry air, that is why I considered 4000 grams, which is 1 kilogram. So 1 kilogram of dry air is going to contain 34.53 moles of dry air. Now, in order to find the mole fraction of H2O, what you have to do is you have to divide the moles of water divided by the moles of water plus the moles of dry air in the total quantity. So, mole fraction of H2O is represented as the moles of water per moles of air. So, we also need to consider the mixing ratio of this water and the dry air, for which we'll have to add this value that you're getting in the denominator, and this is how you find the mole fraction of water. So, if you get a quantity in terms of specific humidity, you have to first find the number of moles in that, say if you're getting 10 grams, then find the number of moles in 10 grams. Again, the number of moles of dry air in 1000 grams, and then divide this value by the sum of this and this value. So this is how you'll find the mole fraction of water and mole per mole. Again, all of these are in moles, so we get in mole per mole. So if you're converting it to millimoles, it will be 16 millimoles per mole. So this conversion is important. Like I said, we want it in millimoles, mole fraction of water, but the quantity that we are going to get will be in terms of gram per kilogram or gram per meter cube. So under such condition, in order to convert the specific humidity to mole fraction, this is how we will do it. This is not the, yeah, mole and mole will not cancel out. Mole and mole, this is moles of water and moles of, that is why we call it a fraction. So moles of water to moles of air. So physically, if you look at, it's two different quantities. And a fraction means it is dimensionless. But although we write it moles per mole, because we are representing the numerator and denominator of two different quantities. How much of moles of air, moles of water is present in how many moles of air is what the representation mean. If you look at the unit, mole fraction is dimensionless. So based on this conversion, which is, I think, something very basic that we have learned during our school days. So we'll be applying this now here. I will again send you this psychrometric chart in the chat. Please estimate this problem that is given over here. Let me just. So I have put in this, I have send the psychrometric chart in the chat box. So using that, I will need you to solve this problem. That is to find the mole fraction of water vapor at 60% RH and 25 degrees Celsius, and also find the saturated mole fraction of water vapor at this temperature. So we had learned how to read the psychrometric chart in the last class. I don't know uh, if anybody doesn't know, you can actually wait for the solution of this problem. I will again say how it is done. So please work on this problem. I have put the psychrometric chart in the chat box. Please find the mole fraction. This is given in grams per kilogram. So please find out the mole fraction. The molar mass of H2O is 18 grams per mole, and M dry air is 28.96 grams per mole. So using this data and using the psychrometric chart, please find out what is the mole fraction of water vapor at 60% RH and 25 degrees Celsius. I hope some of you are trying. Thank you, Ryan, for attempting it. Has anyone else tried to do it? Okay, so I got two answers. Let me move on to the solution. So, okay. So for those of you who weren't there, when I was taking how to read the psychrometric chart, we see the psychrometric chart. I said these horizontal lines are the ones that are giving us values of the humidity, the actual humidity. So corresponding to this, the temperature that I have given here is 25 degrees Celsius and the relative humidity is 60%. So these curved lines are the ones that are depicting the uh, relative humidity. Horizontal lines are the ones that are depicting the vapor pressure or the absolute humidity, and the horizontal line at the bottom, the axis, x-axis, is what is giving us the temperature. So at for this temperature 25 degrees Celsius, this line is intersecting with which curved line? The one that is representing the 60 percentage relative humidity. It is intersecting the vertical line at 25 degrees Celsius at this point. So when we draw a straight line that is projecting to this y-axis, we get around the value of 12. So 12 is the vapor pressure in terms of specific humidity, 12 grams per kilogram. So first we'll calculate what is the moles of water in 12 grams. That is 12 divided by 18, around 0.667. Moles of dry air and 1000 grams of air. Again, it is 34.53 that we already estimated. Now, the actual vapor in mole fraction will be this 0.667 divided by 0.67 plus 34.53, which is correctly similar to the answer that you gave. You should be careful that you represent it in units of millimoles, that is 18.9 millimoles per moles. I had also asked you to find out the saturated vapor pressure, so you can try doing that. Now you've got the actual vapor pressure. Try finding out the saturated vapor pressure in mole fractions. You have the relative humidity, you have the actual vapor pressure. So what is going to be the saturated vapor pressure? So Ryan and Aisha, both of your answers are correct. But again, we are doing a lot of things with the units. So always mention the unit, whether it is moles per moles or millimoles per moles. The answer that you have got it is in moles per moles. And when it comes to water vapor, generally it would be safer to express in terms of millimoles of water per moles. Yeah, yeah, just a second. I'll Harita, can you check if it is visible now? So the answer for the actual vapor pressure is 0.0189 moles per mole. How will you find the saturated vapor pressure at 60% RH? Not very. So here we got this 18.9 millimoles per mole. We know that RH is equal to actual by saturated into 100. Since we have RH, we are going to divide the actual by the RH into 100, which will give us 31.5 millimoles per mole. Now let's cross verify it if it is equal to the value in the chart. So we got 12 grams per kilogram. That is what is giving us this 18.9 millimoles per mole. So if you do the same substitution here to find the actual vapor pressure, yeah, it is 0.0313 moles per mole. Uh, your answer is right, Mayank. So we want to cross verify if this again this 31.5 is because I'm expressing it in terms of millimoles. Whatever answer you will get initially will be in moles per mole. So if you have to convert it to millimoles again, you have to multiply it with 1000. This value by using this formula RH is equal to what is the formula? RH is equal to actual by saturated into 100. The actual is 12. Saturated, we don't know. So we bring RH over here. It will be 60. So 12 by 60 will give us 20 grams per kg. So if you trace it out to in the chart, we'll see that at 25 degree Celsius and 100% relative humidity. It is at 100% relative humidity, the saturated condition will exist. It is around 20 grams per kilogram. Yeah, correct. 9 point. Yes. So right. So the answer is correct. This when you get it in millimoles, you will get this answer. When you get it in grams per kilogram, you will get 20. And then if you cross verify it with the chart also, if you just see at what point is it intersecting the 100% relative humidity, you can see that it is at 20 grams per kilogram of dry air. So again, further from here, we'll be doing some problems. So does any one of you have any difficulty with the psychrometric chart reading or anything? Because otherwise, you will have to uh, waste a lot of time not knowing what to do. If you want me to explain, I can explain again. Otherwise, we'll move on to the next problem. Okay. So then let's move on. So from this, we have understood that transpiration rate is expressed in terms of millimoles per meter square per second. Net assimilation rate is in micromoles per meter square per second. Mole fraction of water inside the leaf is in millimoles. Outside the leaf also in millimoles. Carbon dioxide concentration in micromoles per moles. Stomatal conductance, both of it in moles per meter square per second. So these are the units of the quantities for these two equations. Now when now you can see that how complicated it is. One term is in one unit, this term is another unit, this term is in another unit, this term is another. And at the same time, these two processes, these two stomatal conductance is one process. It is the opening, closing of stomata. But the opening will result in the rate of one gas to be entering at one particular rate and the other gas to be entering at another particular rate. So all these processes as intrinsically it is interrelated. It's very complicated, and this is what we make use of models for, so that all these conversions and everything, it is implemented into this model so that we don't have to bother about all of this. This is just a theoretical understanding behind what goes on inside the model. Even in biocro also, when we give units, we give in terms of we give in terms of general units. All the conversion happens within the model, and the final answer we get is after all the conversion has been done. Now we will do a problem here where I have given the transpiration rate, the relative humidity, the temperature, the carbon dioxide concentration inside and outside the leaf, molar mass of water and molar mass of dry air. What you need to do is find the net assimilation rate. So we have these two formulas, right? We have these two formulas, So using these two formulas, you need to find out what is the assimilation date with this given data as along with using the psychrometric chart. So for psychrometric chart, you have been given the RH and the temperature. So find the actual vapor pressure, saturated vapor pressure, transpiration rate is given. So you will have to find the stomatal conductance for water. Then after finding the stomatal conductance for water, you have to convert it to the stomatal conductance for carbon dioxide. Then use the concentration of values given here to find out the net assimilation rate. Is this question clear to you, everyone? Okay. So this will take a while. Please try to do this. Once you do this, I think the concept of unit and everything is going to be pakka inside your heads. So you can start doing that now. As and when you get the values of whatever, you can post it directly to me. Kindly try not posting it as a message for everyone so that everyone else is also able to, stay inquisitive and curious about what is the answer they're getting. And maybe if any mistakes are done, we can figure out where you guys have gone wrong. So start working on it. First, you'll need to find out the actual vapor pressures from the psychrometric chart. Mayank, can you put in the absolute humidity or specific humidity value that you got in the chat box? Yeah, it is closer to the answer only. You're on the right track. The answer that you gave is the stomatal conductance to water, right? Mm, mm. Ryan, transpiration is equal to GSW into vapor pressure deficit. So the value of transpiration is given. What you have found out will be the value of the stomatal conductance of stomatal conductance to water vapor. Now you have to convert it to the stomatal conductance to carbon dioxide and then find out the assimilation rate. Should I wait for more time or shall I move on to the, if you need more time to solve it, we can, um, and once you respond only, I'll be moving on to the solution. So, yeah, okay. Okay, I'll be waiting for some more time. There are people solving, so let them finish and then I'll go to the explanation. Yeah, right, correct. So the only variation in my solution would be the absolute humidity that I took. Uh, so since we are taking it from the chart, it will be a little bit approximate. And I think I took values so that the calculations are a bit more easier. Let's go to the solution. So for relative humidity 50 percentage and temperature at 30 degrees Celsius, we're getting around 13.5 gram per kilogram. Ryan, what is the value of absolute humidity that you considered? Let's give it here. 13 gram. Okay, so no problem. The Okay, no problem. So the answers will be the decimals and everything will be a little bit different considering the value that I have taken here. But I think in the end, whatever answer you're getting is right only. So it is 13.5 from this chart. And next, what we'll do is we will convert it into mole fraction. So to convert it into mole fraction, we divide this by 80. So we get 0.75 moles. and moles of dry air is 34.53. So the actual vapor pressure in terms of millimoles per mole will be around 21 millimoles per mole. I think you got it around, Ryan has got it at around, yeah, 20.48, which is, yeah, correct. Even Mayank also got something like that only. I think Mayank, so what did you get? You given the GSW, not the VP. So this is the actual vapor pressure or vapor pressure in millimoles, mole fraction that you will get. Now next, we have the RH. So instead of taking from the chart, I directly estimated using this formula. So 21 divided by 50 will give you around 42 millimoles per mole. So this is the actual, the saturated vapor pressure. So the vapor pressure deficit will be 42 minus 21, around 20. I think even you got that. Uh, yeah, somewhat the same. Again, one point something and point something differences will be there considering the number of decimals we are taking and all the rounding off. So I think Ryan, you got the correct answer. Let me see Mayank's. So we got the vapor pressure deficit. Now we know transpiration is GSW into the vapor pressure deficit. So we will divide, since we know the transpiration value, which is 5 millimoles per meter square per second, we will divide the GSW by 21, which is the vapor pressure deficit we get, and I got around 0.24. I think, yeah, even you have got it right. Mine 22.4 millimoles per meter square per second. Okay, it is supposed to be in the units of moles per meter square per second because I told stomatal conductance will be in the units of moles per meter square per second. It is only then you can use it in the assimilation rate because assimilation rate you're getting it as micromoles per meter square per second. So if you substitute the millimoles per meter square per second, in the end you will have to do some conversion in order to standardize the unit in the left hand side and the right hand side. So that is why the stomatal conductance is always going to be in units of moles per meter square per second. So here the conductance to water is 0.24. So carbon dioxide diffusion is slower than this water. So we will divide this value by 1.6. So we get around 0.15 moles per meter square per second, around 1.5 times lesser than the water's value. And that value we will substitute in this GSE part. We know that the carbon dioxide concentration outside the atmosphere given as 400 and inside the leaf is given as 250. So doing this substitution in the end, we will get 22.5, around 22.5 micromoles per meter square per second. So yes, your answer is right, Ryan. Mayank also has done some of the correct thing until some point, after which I think you got a bit confused. So what we have to remember from whatever I taught today is about the units, transpiration in millimoles of water per meter square per second, assimilation in micromoles per meter square per second. The mole fractions of water and carbon dioxide will be, water will be in millimoles, carbon dioxide will be in micromoles per mole, and stomatal conductance is always in the unit of moles per meter square per second. So it is only then that this same stomatal conductance value by connecting it to the diffusivity ratios, we can substitute, we can consider for the transpiration rate as well as the assimilation rate. So this is how this entirely connected this. So this we are calculating it for one particular point, the relative humidity, the temperature, all these are varying values. So when we consider a model the model is accounting for all of these estimations for one entire day or for one entire hour, depending on what are the varying conditions of humidity and temperature. So for 1 hour, maybe the humidity is 60. Humidity doesn't change on a hourly basis to a great extent. But say if it is the 12 noon and something around going to 1 o'clock or 2 o'clock or 3 or 5 in the evening, the humidity value changes, temperature value changes based on this, all of the stomatal conductance and transpiration rate and assimilation rate, all of these changes. So this is how we represent the physiological processes in terms of equations and in terms of numbers with the help of all of these estimations. And the Biochrome model is also doing something like this only, that too at a more complex way by doing estimations for every hour, not just for this one point, but every hour. What is the humidity? What is the temperature? What is the light obtained? What is the carbon dioxide concentration? Based on this, the calculations are done for every year, and this hourly calculations will be done for an entire season, say for 2 3 4 months, and that is how we will get the net uh, photosynthetic capacity of that plant. Or if we want to do it for a particular leaf, we can also do that. So this is just a gist of how the estimations are done by the model. So this will be it for today's class. Tomorrow's class we will be again going into models that are balancing this carbon. There is a model that is used in Biochrome, stomatal conductance model. So I'll be talking about that in the next class. For today's class, this will be it. Um, if you have any doubt, I'll be here till 4:30, or you can use the Google form. Otherwise, you're free to exit the session. Thank you. Thank you for attending today's session. Aisha and Shreyas, if you have, do you have any doubts?